So now we come to the final lecture of this session of rare and neurological disorders. This will be by Professor S. Ganesh, who is going to speak on glycogen in neuronal life and death. Professor Ganesh is presently Professor of Biological Sciences and Bioengineering at Indian Institute of Technology, Kanpur. He is Dean of Research and Development at IIT Kanpur and has served as staff scientist at Riken Brain Science Institute, Japan. Professor Ganesh is an elected fellow of the National Academy of Sciences, India. His other honors include Outstanding Young Scientist by Scopus, National Bioscience Award for Career Development, and Birla Science Prize for Young Scientist in Biological Sciences 2009. He was also awarded the Outstanding Research Investigator Grant by the Department of Atomic Energy, the CDRI Award for Excellence in Drug Research, Rajiv Goyal Prize in Life Sciences, and the Ramana Fellowship DST. Please welcome our speaker, Professor S. Ganesh, Glycogen in Neuronal Life and Death.
Good afternoon to everyone, and let me begin my talk by thanking the organizers, especially Dr. Thangaraj, for inviting me to give a talk in this symposium or workshop. What I would like to do in the next 20 minutes is to give a glimpse of why and death of the neuron. If you look into the neurodegenerative disorders, there are a large number of inclusions that are known, which includes, for example, the A beta or the tangles, or in case of polyglutamine disorders, there are aggregates, and so on and so forth. But what is very interesting here is that there are a group of disorders wherein the inclusions are not protein, but they are an abnormal form of glycogen called as polyglucosan. And such inclusions are called with very names, like there are, for example, in aged brain, they are known in a different name and so on. But all of them are common in the sense that they represent one or the other form of glycogen. And our lab has been working on a disorder which is Lafora disease, again a polyglucosan body disease. And in fact, this disease led to the identification of a protein called laforin, which has a carbohydrate binding domain. And in fact, there are only two such proteins that is known in the human proteome, which has the ability to go and bind to the glycogen. And it also has the phosphatase domain. And there are studies that have shown that this protein binds to glycogen and dephosphorylate, and the significance of which is not known. But what is known is that many of these inclusions that you see in abnormal conditions are rich in phosphate content, and whether that relates to anything that a kind of a housekeeping role that this protein has is something that is questions being addressed by many. These inclusions, you see that in many conditions, especially in the Lafora, you find this inclusion all over the brain. What I'm showing is a mouse model, and it's true for even the human, that the inclusions are everywhere, which sort of represent the density of the neurons that you see, showing that they are very, very specific to the neurons, and then you present it everywhere. And our group has done a lot of work on this to understand how the disease, what are the mutations there, and then how these defects affect the way the protein function, and at the cell level, tissue level, and organism level, how it may contribute to the disease symptoms that you see, and so on. And we sort of propose certain models using which we are now trying to fix some of the symptoms using mouse model. And one may say that these are abnormal conditions unique to a particular rare disorder. Therefore, the question still remains as to what is the relevance of glycogen in the neuron per se. The reason being that normal neurons do not store glycogen. Because most of the energy they derive from the astrocytes. But what is interesting here is that if you look at the neuron, they still have the machinery. For example, the what you call as muscle glycogen synthase, the critical protein that is required for synthesis of glycogen, is expressed even in the neuron, though you don't find glycogen in the neuron. Therefore, the question is, why should neurons have glycogen synthetic machinery? That's the question that we address. That's what I'm going to share with you today as to why neurons possess this machinery, though they don't store glycogen in normal conditions. What is interesting here is that there is one paper that came from Gunavat group in Barcelona, which showed that if you delete glycogen then in fact it sort of help the animals, especially animals, flies. Here what is shown here, first let me put this paper first. Here what is shown here is if you overexpress the constitutively active muscle glycogen synthase in the neurons of the fly, then you see that the flies they don't live longer as compared to the wild type. Their lifespan goes down, survival is affected. That's the case even with the mouse if you sort of express it in the neuron. It shows that if you forcibly activate the glycogen synthase in the neurons, it affects the survival of these animals, and maybe the same is the case in case of humans, because there are conditions wherein you do see glycogen, and as a result the individuals die early, the Lafora being one of such conditions. The other paper from the same group, from Gunevert's group in Barcelona, what it showed is a very interesting thing. But if you knock down RNA-mediated knockdown of glycogen synthase, and you see that fly live longer, then if that is beneficial, and of course you can also knock out the glycogen synthase in these conditions, and then you can find that many of the markers that are there in the mouse brain that are aged, it's about 16-month-old animals, where you have accumulation of HSP70, advanced glycation end product, and synuclein. Such aggregates also disappear if you knock down the glycogen synthase, suggesting that not having glycogen synthase probably is good for us to live longer. The question is, why should neurons have these machinery in them still active? We started with a simple hypothesis. Therefore, what I'm going to show is more of a functional study, and then we'll model some neurodegenerative disorders to address this question. Our hypothesis was that glycogen is toxic to the healthy neurons, but it is synthesized by the neurons when they are under physiological stress as a maybe a transient cytoprotective role probably they play. When I say glycogen, I also mean the glycogen synthetic machinery. Glycogen could be a byproduct. That is something that I would address in my talk. So this is what we simply started with, a cell line model. These are the neuronal model, but we also done with the primary neurons. But what we have shown here is that if you expose the cells to one or the other form of the stress, for example, proteasome blocker or oxidative stress, then neurons synthesize. They do have some very, very low level of glycogen that goes up in these two cell lines. But such kind of things don't happen if you look at the non-neuronal cells. I'm just showing you data for one cell line, but that's true for many other. And that's just to show that the pink staining that you see are the glycogen. Here we use a different model wherein we've overexpressed, for example, the wild type form of Huntington mutant, form of Huntington. The other one is the expanded Huntington, which is known to be cytotoxic. When you express them, again you find that neurons synthesize glycogen, possibly as a response mechanism. That's true with even fly. These are brain sections from the fly that stained for the glycogen, pass staining. And you can see this is a GFP, and there's a GFP with Q20. But if you overexpress the Q93, which is the disease-linked mutant allele, then you would find that the level goes up. You can see the staining pattern, but you can also measure with time as well the level of glycogen in the neuron goes up because they're all driven in the neurons, right? And it clearly suggests that possibly the stress induces the glycogen synthase to synthesize glycogen. And you can see that even in the Huntington mice brain, this is from transgenic animals that have overexpressed the Huntington mutant allele. Again, you can see that the glycogen level going up. You can measure that and show that the glycogen level goes up. The question is, why should? What is how does the glycogen go up? And that's also we dissected. Of course, we know well that the glycogen synthase is the one that we looked at the phospho form of it, which is the active form. And then you can see that as you express the Huntington protein here, for example, the phosphoform is inactive here. So you can see that the level of that goes up, suggesting that this is the activation of the glycogen synthase that leads to increased glycogen. And we could see that even in the Huntington mice, for example, in the Huntington mice 2-month-old animal, the level of the phosphoform goes down, suggesting that there is an activation of glycogen synthase. Why should then neurons under stress activate glycogen synthase and synthesize glycogen? That's what we asked. And we looked at first the localization of these aggregates, very, very dramatic thing that you can see here. For example, these are the aggregates that you see in the neuronal cells, but if you overexpress with a wild type form of the glycogen synthase, you will find that majority of the cells, these aggregates sort of disappear. They become more or less uniform cytoplasmic staining, suggesting that somehow this glycogen synthase activation helps in reducing the aggregate formation. What we have done here is that we have overexpressed another protein called R5, which is also a factor that is known to activate glycogen synthase. So we have not overexpressed the glycogen synthase, but we overexpressed an activator that also results in very similar condition that you see here. And you see that the aggregate goes down, and we confirmed further by knocking down the glycogen synthase. If we knock it down, you'll find that the aggregate form, which is stacked, trapped in the stacking gel, as well as the total level of these Huntington protein goes up if the neurons lose glycogen synthase activity, or partially when it loses. That is what's shown here. Conversely, if you overexpress them, glycogen synthase or the R5, which again activates the glycogen synthase, you see that the aggregates pretty much disappear, suggesting that glycogen or the glycogen synthase somehow help the cell to clear these aggregates. We tested if that is the case, then the neurons should have a lesser amount of these aggregate cytotoxic protein. Therefore, you should really help the fly. This is a fly model. You can see here, for example, when you have the Q93 expressed along with the human muscle glycogen synthase, they tend to live longer as compared to having only the Q93, suggesting you certainly have some protective function when you overexpress. So how does it do that? What we have found is that the glycogen synthase activation results in the activation of autophagic pathway in the neurons, and that happens only in the neurons. In non-neuronal cells, the glycogen synthase is not that critical for autophagy. What we found, it's a busy slide. What I would like you to concentrate on these two sections. Here we have overexpressed the polyglutamine, and then you have along with either the R5, which is an activator of glycogen synthase, likewise here with the glycogen synthase itself. And in both the case, what you'll find that you see these LC3 markers and the P62 suggesting that there is an induction of autophagy when you overexpress the R5 or GS, which in turn activate the glycogen synthase. And that is shown further by these assays. Conversely, you can knock down the glycogen synthase, and you'll find that autophagy is compromised, suggesting that glycogen synthase is very much required for the neurons to activate autophagy. And we have done that using looking at the transcripts. For example, if you overexpress the R5, which activates glycogen synthase, many of the transcripts of the factors that are involved in autophagy go up, suggesting there is an activation. And if you knock down the glycogen synthase and try to induce autophagy and see what happens to their level, it remains low, suggesting that glycogen synthase is required for the induction of autophagy in neuronal cells. And therefore the question is that why should overexpression of the muscle glycogen synthase in the neuron and the mouse should have an effect which affects their survival? That is the question. What is interesting is that when you overexpress even glycogen synthase or the activator of glycogen synthase, what you find is that the neurons die. In other words, normal neurons, which are not under any physiological stress when you are culturing using optimum condition, and if you overexpress them, then they die. it is not good for them when you activate glycogen synthase when the cells are under normal condition. However, if you expose these cells to if you expose these cells to some of the blockers of autophagy, then what happens is that this cell survival is restored, suggesting that induction of autophagy in a normal condition is not good for the neurons. Neurons really cannot tolerate if autophagy is induced when the neurons are under normal condition. So probably that is very reason why the glycogen synthase is not active when the neurons are happy. It needs to be activated only when they are under stress. That is just to show that you can prevent the autophagy. It is not just linked to the glycogen synthase. You can even mimic that using rapamycin, well-known activator of autophagy. So if you add rapamycin to the neuro-TVA cells, you see that the neurons die. However, you do the same in the presence of, for example, bafilomycin, then they restore the cell death, suggesting it is the autophagy induction that kills the neurons when they are in normal condition, right? So that probably would explain as to why the fly, which are happy otherwise, if you overexpress the glycogen synthase, they die, could be because when the neurons are not under stress, if you activate glycogen synthase, that is detrimental to the neuron. So we did interesting study. So what we did was we took these flies which overexpress the muscle glycogen synthase and then exposed them to the, for example, hydrogen peroxide, extra distress. And then you find that, and then we sort of scored the mortality. And you can see that if you have muscle glycogen synthase overexpressed in the neuron, and when the flies are exposed to certain stress condition, they can live better. And if you knock down the GS, and then their survival goes down, suggesting that these proteins are required when the neurons or when the animal or the neurons under some sort of stress. That could be true because even we looked at the human brain samples that from the postmortem brain samples from Alzheimer's, Parkinson's, and Pick's, and many other, and these are just SOD markers to show that there could be oxidative stress. In all these conditions, you will find the accumulation of glycogen, suggesting glycogen synthase was activated here, but the same age-matched normal brain of, again, individuals that are dead because of some other reason, accidents and so on, you will not find to that extent the glycogen being there, suggesting, again, these are linked to what kind of conditions that the neurons are exposed. So that is just to tell you that glycogen synthase is required for the induction of autophagy. Then the question is about glycogen. So is it that the glycogen, a product resulting from the activation of glycogen synthase just for activation of the autophagy, or even the glycogen per se has got a role? That's what we looked at in our next set of experiments. What is interesting here is that when you go stain the cells that express the polyglutamine protein, you find the glycogen synthase localized around these aggregates, okay? And that's very interesting. And second, we also stained for the glycogen. There are antibodies available, and you'll find that often, you'll find the glycogen particles being around these aggregates, suggesting that, even the glycogen may have something to do with the preventing the aggregation. So in collaboration with one of our colleagues, what we did was we looked at the aggregation kinetics of pure peptide having 35 glutamine. So if you add the glycogen, pure glycogen, and what you see is that then the kinetics it goes down. Somehow it is able to delay the aggregation propensity of that peptide, suggesting that maybe a kind of a crowding effect that it offers. Maybe that could be a reason why neurons may synthesize these glycogen. And this is again to show that whether glycogen even in vivo condition, in neuronal cell lines, whether glycogen has a role. So what we did was we did a knockdown of the glycogen synthase, but activated, for example, the autophagy by using rapamycin. Therefore, autophagy is not compromised to that extent. And we looked at the aggregates, whether if you activate autophagy, but prevent the glycogen being there in the neuron, whether still you find the aggregates, and you'll find that it is better than this, but still you find the aggregates being there, suggesting that glycogen probably has some role to play even in preventing the aggregates and helping the neurons to get rid of such abnormal species and degrade them. And possibly that's the reason why glycogen synthase is recruited nearby to make the glycogen in the vicinity of the aggregates. That's just suggestion that we don't know. Just to summarize, what we believe is this is the hypothesis that we started with, that glycogen is toxic to the healthy neurons, but synthesized by neurons under physiological as a cytoprotective mechanism. Therefore, it has both a good function and a bad function. So bad is when it is activated in conditions when the neurons are normal, but good is when it is activated when the neurons are exposed to certain stress, and that by inducing autophagy they are able to protect. So that's the kind of finding that we have. And just to thank this study, this is something that we started very recently. This was done mainly by Anupama and Akansha. Akansha is still continuing her study on this in the fly part. And I would like to thank my collaborators, Ashwini for the peptide aggregation studies, Nihar for the Huntington mice, and Professor Shankar from NIMHANS for all the brain samples and so on, and then of course the funding bodies for helping my research. So that's from me. I'll be happy to take any questions here.